Hello everyone. I'm very pleased to meet you today and for this special webinar regarding the testing trends from New York SCC Suppliers Day. And with Ilona, we will give you some insight regarding this testing, especially for the North America. And also we are really delighted to have today with us some companies that you may know, but they will also give you their insight and what is very important at the moment, what are the novelties, what are their trends. So we are delighted to invite the company from, we will start with David Suzat from Biome Labs, then with Mr. Moura from CIDP from Brazil, and then we will go with Mr. Jonas from CBAS with the Nevisance instrumentation. Then we will go on with Guillaume from Wineos regarding solar sun care protection, and Arthur from Loretta will deep dive in the atomic force and the tribology and typology of corneocyte. So let's go. We start with David. So.
Good morning or good day, everyone. Sharing my screen. I don't have any feedback. Is it okay on the screen?
Yes.
Okay, perfect. So hi, everyone. I'm going to talk about microbiome. So the microbiome is all the microorganisms, bacteria and yeast that each one of us we have everywhere in our body or on our body. I'll be more focusing on the skin microbiome, but at Biome Labs, we are specialized in skin, scalp, oral, and vaginal microbiome. We are a biotechnology startup specialized in testing and analyzing the microbiome for the cosmetics, dermocosmetics, and pharma industry. We are a team of 17 people, mostly scientists, engineers, and technicians in all the fields that are required to analyze the microbiome according to the way I'm going to explain it to you. So microbiology, immunology, genetics, and artificial intelligence. Concretely, microbiome on the skin or on the scalp are all the microorganisms that we have, and the microorganisms are the link between your skin and the product that you have on your skin. And so this microbiome opens a new field, allowing to have the possibility to recommend the best product specifically suited for your specific skin. So that's why the way we work on the microbiome at Biome Labs is what we call the MRI of the microbiome. First, we test what we call the impact, so to know how your ingredients, your formulas, or your products impact the microbiome. Then we devise a specific fast on-site measurement kit to be able to measure within 2 minutes the specific microbiome of a consumer. And then we've got some AI algorithms to find the best match between your products, which have been previously indexed in the database to know how it's behaving on the microbiome, and the specific consumer microbiome. Alors, we've been, so the company is 2 years and a half now, and so we've had many customers. Some coming from the US, so either at the New York Suppliers Day or at the CES Consumer Electronics Show in Las Vegas. And in terms of best practice, what we've seen in terms of uses about the microbiome and the requirements that we have are summed up on this slide. First of all, there is a need for consumers to know their skin, measure their skin, and to find the best match about which product will be the most compatible for their specific skin. Another use of this technology is to be able to follow along within time the evolution of the skin and the evolution of the microbiome of the consumer by fast and low-cost microbiome clinical measurement, even every day. This technology allows to test your ingredients and your products to see if they respect the microbiome, and so to be able to claim to the consumer that the product respects the microbiome. Some other use of this technology will be to guide the formulators or the brands to formulate products specifically suited for a given consumer microbiome or a given type of consumer microbiome, and of course generate a lot of data to be able to know more and have more data and more insight about what's going on the skin of the consumers. If I deep dive a little bit and very fast on the way it works, products do have, even if they have a similar in key list and similar ingredient, they do have some very different impact on the microbiome. Just a little change in the formula can have a quite large impact on the microbiome. Here we illustrate with different moisturizers from the European market and the way they impact the microbiome. You can see that there is a quite large difference. Then to measure and to know the specific microbiome of the consumer, we've devised some tests which are extremely similar to COVID antigen tests, but for the skin or for the scalp. So basically, there is a specific sampler system to be able to get your microbiome on the skin, which even works when you are maked up. Then this microbiome is placed on strips on which you find different antibodies. Each antibody is able to measure in a semi-quantitative way the one specific parameter that you've got on your skin, for example, acne, atopic dermatitis, and so on. To read the results, like a QR code of your skin, you've got an app that the BA, the pharmacist, or the physicist can download on their tablet or on their smartphone. They take a picture of the disc here, and instantly you get the results of the microbiome of the user. And then our AI algorithms recommend the best match between the products which have been previously indexed from your brand and the specific microbiome from the consumer. Just as a fast illustration, it's working like that. So it's a co-branded product where you've got a sampling system, a system to get back the microbiome, and then the strips to read the microbiome. Time is passing by extremely fast. I've got 1 minute left, if I'm not mistaken. So in terms of AI, we recommend the best product with specific algorithms, so biological and microbiological algorithms that we've developed with what we call an impact score. So to illustrate that, here, you've got on the horizontal axis some people with their specific microbiome. In the vertical axis, you've got the compatibility of the product with the specific microbiome. Green, it means it's very compatible. Red, it's less compatible, even not compatible. And then you get here the sensibility. And when you've got some points here, you can see that one same product, which are the blue dots that you can see here on the screen, one same product can have very different compatibility. It can be in the green for some consumer and in the red for some other consumer. The question is, as a consumer, when they want to buy a product, which is my skin? How do I measure my skin? And which are the product compatible specifically for me? This solution allows to answer that. From a front-end perspective, so from the beauty advisor perspective or the physicist or pharmacist, there is an iPad app. You just take a picture and you get the instant reading of your microbiome, and then the recommendation of your products ordered by the most compatible first to your skin to be able to find your specific beauty routine. Voilà, thank you very much. If you've got some questions, I'll be happy to answer. Thank you so much.
Thank you very much, David, for this presentation. Just for the participants, you can put your question on the Q&A in the app of Zoom, and then we will answer later at the end of the webinar, or everyone can also answer if it's concern a special field. So now I'm pleased to introduce Mr. Moura from CIDP from Brazil.
Hello, it's a pleasure to meet you and talk to you and Charpentier and all the audience worldwide watching this incredible webinar by SkinUps. Let me share my screen with you so we can discuss a little bit on our experience on hyperpigmentation. In a second. Are you able to see my screen? Good. Hello, so today I'm going to be talking about the clinical methodologies we use in hyperpigmentation studies and how these can be used to substantiate claims in the clinical research involving cosmetic products. My name is Renato Moura. I am a dermatologist, member of the Brazilian Society of Dermatologists, and currently a director for the CIDP Brazil. Me and the team of 7 dermatologists in Rio and others worldwide are able to run clinical investigation studies in vivo for efficacy, safety, and tolerance evaluations. We, as CIDP, are a full-service CRO, and we offer from preclinical trials to clinical studies, including medical writing, and we are located in 4 centers worldwide. So just to highlight a few concepts that are common in all of hyperpigmentations, we are talking about the melanocyte, our main cell in deposition of melanin. This is located in the basal layer of the epidermis, and currently we see that there's a density of about 1,200 cells per millimeter square of skin, and that is constant across all skin types. What you see in the different phototypes is this deposition of the melanin is different in terms of the vesicles that are formed and the amount of melanin that is deposited into the keratinocytes that are surrounding the dendritic projections of the melanocytes. Here on your right, you see an example of a brown melanocyte that is delivering these melanin granules on top of the nuclei of the keratinocytes in order to provide a cap protection against UV radiation. And what we see in hyperpigmentation lesions is that the melanocytes actually are not multiplied or in numbers differently than the normal skin. Instead, what you see is a hypermetabolic state where they are growing, enlarging, becoming more dendritic, and eventually delivering more melanin to the cells in the basal and upper layers of the skin. So just so we highlight our main objectives in clinical studies is to see pigmentation as 3 main axes, which are the solar lentigines in the first place, which are commonly known as sunspots or solar melanosis, and they are caused by UV radiation, the PIH caused by acne inflammation, and the melasma, which we're focusing basically in epidermal superficial melasma. So for solar lentigos, what we are seeing is usually those very well-demarked macules that are seen on the prominent areas of the face, and they are permanent lesions that do not go away or do not improve when there is lack of sun exposure. And they are also related to the solar elastosis and the degradation of the collagen on the dermis. And they are more, they can improve in their color and their treatments, but not in size. And what we generally see these lesions are much more persistent than the other ones that we are usually following on this one. Here on the right, you see a solar lentigonal macro photo and the image of a microscopic augmentation zoom, where you see all the clinical characteristics of one that we dermatologists analyze when we are watching the skin. So the sunspots can be looked in a more diverse way and in a more global way. We can run clinical scales that will be useful to quantify the changes over time, and with the use of the 10-point scales with statistic validation of the medical evaluation, you can achieve higher replicability and reproducibility in your assessments. On the second type of marks that we are usually targeting are the ones that are related to acne inflammation, and that are more common in phototypes 3 to 5 or in 6, and they are more likely to present PIH instead of PI erythema. And there's a direct relationship of these lesions formation with the size and the manipulation picking the acne itself. And direct measuring methods are suitable for these kinds of lesions. But of course, in an ever-changing lesions of acne in the skin, you may see some limitations, including the onset of new lesions near or on top of the previous lesions that we are following. In the lower case here, you see lesions reducing in size, but also improving in contrast against the surrounding areas. Here, we also following how these lesions change differently and how we can use different image techniques to show you the improvement of the lesions. On top, you have standardized polarized images, and down on the lower layer, you see that the lesions are improving, and on a microscopic level, this change can also be quantified. We also have other tools to analyze and to quantify this improvement over time. And usually we are counting on global improvement, where we are comparing against baseline using standardized scales, the severity of the lesions and their heterogeneity against the background can be assessed through the PIHASI, which is an area score that will have the lesion severity scoring after all, and the homogeneity of the distribution as we've seen in global evaluations. The size of the lesion in this case can be followed with more precision, because these lesions tend to improve in size because of the characteristics of the fading outside inflammation. And the intensity of this pigmentation can be followed through absolute standardized color scales. Here we have an example of some scales that we use in the clinical practice, and they are useful to show and to analyze on each time point the evolution of the hyperpigmentation across all Fitzpatrick types. So for instance, when we apply this methodology here, you're able to visualize and to quantify the amount of change that you see in every lesion. And in this case, the two first lesions, they enhance differently, they improve differently than others. So these methods are able to capture these differences in intensity and contrast against the periphery, and we are able to generate our results. Here we have another example of lesions clearing over time and improving in terms of pigmentation and also erythema. For the melasma, we have specific situation. Melasma is actually a condition that is very chronic on the skin, and that is very responsive to sun exposure. We usually prefer studies when we can compare the Kligman triple formula as a gold standard in the topical monotherapy compared to a cosmetic treatment, for instance. And ancillary sunscreen, of course, is recommended for all subjects. And to differentiate the effect on the control group would be useful to have a comparator, like a study design like this would provide. In our clinical practice during the inclusion, we are carefully selecting the melasma that allows for the optimal results. And these are the epidermal melasmas, the ones who present more superficially, and that the wood light and the UV light can, for instance, highlight these lesions. When we apply the UV light, the lesions themselves become more contrasted against the background. And this is why we're able to follow these kinds of lesions and see their improvement. What you see here is also the selection also has to do with some characteristics of the melasma. When has it, when was the onset of it? Was it related to pregnancy or not? And the area itself might also show some features that are very important. The lateral kinds and mandibular kinds of distribution are more related to a very chronic sun exposure and very hard to treat melasmas. And in order to analyze the melasma, we have different light modes that we can apply, including the cross-polarized, which will highlight the contrast and the tone hues of the spots themselves. The ultraviolet cross-polarized light is also very important to deepen this light and to be able to see deep inside the dermis for the pigment changes over time. Here we are also applying in our studies our own melasma atlas that we have created in order to assess under the M-MASI, which is the modified MASI score, and we are applying this to our Fitzpatrick phototypes. It's important that we have referential images so that the doctors, the investigators, can be able to understand the skin and have a validated perception and a reproducible perception of the pigment changes. Also, we see and we have to analyze the differences across phototypes. So the severe melasma across the phototypes may have different contrast presentations, and the atlases and referential images allow for a more accurate evaluation across the time points. Of course, I would like just to mention that we use the chronometer as a simple instrument that has many applications, and we're able to see these lesions. And for melasma specifically, we have well-demarked lesions that can be compared against the non-lesional adjacent area. These repositioning and these measurements, when they are accurately repositioned and highly standardized, allow for sampling of the color, including the parameters LAB that assess pigmentation, but also erythema. Of course, this method has a limitation on the size of the lesions, and the sampling method is a continuous scale. We need to validate each measurement against the surrounding skin. That was my message for today. Thank you very much for your patience. I think I was right on time. And thank you for sharing this moment with us. Thank you, Anne. Your microphone is off. I'm sorry, Anne. We can't hear you.
You're muted. We can't hear you, Anne.
Sorry. Yeah, thank you, Renato. I think you have a question on the Q&A session, so you can answer directly, and we will answer at the end also, discuss about this point. And now I'm delighted to introduce Jonas Fredriksson for the Nevisance presentation. Thank you.
Thank you, Anne, and hi everyone, and thank you for joining this webinar. My name is Jonas Fredriksson, and I'm with Sybase, and we are a Swedish medtech company based in Stockholm. The title of my presentation is an objective, quantitative, and robust approach to addressing skin barrier function. I'm going to start a little bit to talk about the technique and then moving on to share some data. The technology is based on electrical impedance spectroscopy, or EIS as we call it. On top of this technology, we also apply AI-based algorithms, which help us to gain additional insight from the measurement. And this is not a new technology. The research started back in the late 80s at Karolinska Institute in Stockholm. And since then, the technology has continued to develop and has become a clinical validated platform for skin cancer detection. It's FDA approved and CE marked, and if you go to a dermatology clinic in US and Germany, you can be tested for skin cancer. But when it comes to skin barrier function, we have taken a medical development approach, starting with atopic dermatitis, chronic skin dryness, and are continuing to build our portfolio of indications. And we are now entering the cosmetic field also. And we have a strong body of published research. As you can see here, there are quite a number of publications relevant in the cosmetic context. And the device can be used both in vivo, ex vivo, in vitro studies across different populations, from newborns, pediatrics to adult patients. And EIS works by sending small electrical signals into the skin and measure how the tissue responds. Each measurement takes about 10 seconds and generates 700 data points, so we get a large amount of information from a single measurement. The signals are impacted by skin structure, cell organization, hydration status, and lipid composition. And we are using multiple frequency, which allow us to go deeper and reflect the overall condition of the tissue. And this is why EIS can detect early and subclinical changes even before they become visible. Here you can see an example comparing normal and atypical skin. The curves looks quite different, and which allow us to quantify the difference between skin condition and then translate it to clinical relevant information. So EIS doesn't measure a single parameter. Instead, it provides an integrated view of the skin condition, including these factors as the cell organization, the membrane integrity, tight junction, lipids, hydration, and tissue inflammations. Before I going in deeper into the data, I just would like to touch on the difference with transepidermal water loss. And as you know, the tool measured water evaporation from the surface and indicate the barrier leakage, but it has a quite high variability and is sensitive to environmental factors as humidity and temperature. If you look at EIS on the other hand, it measures the electrical properties of the skin and it reflects underlying structure of the tissue. It's sensitive to early and subtle changes and less impacted by external factors. And because the measurement uses multiple frequency, it also provides information from different depth. It can go up to 2.5 millimeters. And I think together, these two methods, they could complement each other and provide a more complete picture of the skin condition. And tool tells us more about the barrier leak, while electrical impedance helps to understand why it's leaking. This is a quite recently published study that compared EIS with established methods, and it confirms a strong correlation with tool and coronometry. And at the same time, EIS was able to detect early subtle changes that tool did not capture. And you can see what the author conclude there, that together they provide a complementary insight into the skin barrier integrity, and EIS may serve as an early biomarker for skin barrier damage. And here is also a head-to-head study comparison between tool and EIS when it comes to sensitivity to external factors. And in this study, they looked at something as simple as a stair climbing, and within just a few minutes, the tool value changed quite a lot. With EIS, we don't have this issue. We are measuring we are not measuring how much water passes through the skin. We look more at the actual structure of the skin, and it's less affected by external factors. So if I sorry, here, yeah, here. So I would like to share one study on atopic dermatitis here, and this is a study that we perform together with Chesme Actis Group in Switzerland. And we started with a question: can we identify atopic skin even when it looks normal? And as you can see here, there is a clear difference between healthy controls and non-lesional atopic skin, indicated that NEVISENSE can detect subclinical changes, and tool was less sensitive in this study. We also wanted to see if we can detect subclinical changes around the lesion. So we compared healthy, adjacent, and lesional skin. And also here we saw that EIS clearly separated all three groups. Again, tool was less sensitive and failed to separate adjacent from healthy skin. So this reinforced that EIS detects subclinical information. In the same study, we also wanted to see if it could be used to monitor treatment efficacy. And looking at the graph on the left-hand side, we see that the patients show a clear improvement within 3 weeks, and it correlates also with clinical markers as pruritus and SCORAD. And it also correlated with inflammatory biomarkers. So EIS does not only detect barrier damage, it can also be used to monitor treatment efficacy. And this is an interesting study, ex vivo study also, using human skin sample. In this study, the skin was exposed to different concentration of everyday detergents, similar to what you can find in a grocery store. As you can see, EIS show a clear dose response curve, and where higher concentration led to increasing barrier damage. And these changes also correlate with inflammatory markers. Finally, I would like to share some newly presented data also. And the first study here is also an ex vivo study that confirming the result from the previous study. Here, EIS detects barrier damage just within a few minutes after an SDS exposure, but also interesting that it shows a recovery after treatment. The second study here is a study from University of California, San Francisco, that was presented at the SID meeting in Chicago a few weeks ago. And there we can see that EIS detects age-related skin barrier changes with a strong correlation to biological aging. And finally, a study from Mount Sinai on babies with atopic dermatitis, that they could predict the risk of developing atopic dermatitis before onset of symptom. So that was a little bit about some studies, and sorry here, and again, to summarize here, if your goal is now to gain deeper understanding of the skin barrier integrity and what is happening within the tissue itself, I think NEVISENSE offers you a new dimension of insights. And we also believe that this opens new opportunities across skin barrier research, product evaluation, efficacy testing, and claim substantiation. And if you have any questions, if you would like to get any of the publication that I shared here, or if you would like to have questions, learn more, or a demo, just contact me. You have my contact details here, or you can scan the QR code here. I'm also happy to answer question during the Q&A session at the end of the webinar. So thank you very much for your attention, and thank you for having me here today. Thanks. And with that, back to Anne.
Thank you, Jonas. We will wait for the Q&A to ask some specific question. Now I introduce Guillaume from Wineos. He will speak about sun care protection, in vivo and in vitro testing.
Thank you, Anne.
Everything
Okay.
great.
Everything okay? Great.
Everything okay.
Hello everyone, I'm Guillaume Lejeune from Wineos, and I will present you our company and our field of expertise, which is sun protection testing. Wineos was created more than 25 years ago as a laboratory specialized only in sun protection testing. It means that we don't do, for example, hydration testing or anti-aging testing. We are only focused on sun protection testing, which is the heart of our expertise. When we created the lab, we had the goal of making sun protection testing more reliable internationally, and thus by different objectives. In a first way, to develop new efficient methodologies to assess the sun protection parameters, such as SPF, UV APF, or water resistance, for example. And in another side, also to develop new technologies, appliances, to help the industrials, the testing laboratory, and the cosmetic brands to improve their R&D and to improve the testing process for the products. And so over the past years, we developed a lot internationally, and with our expertise, we've, for example, participating into ISO consortium, into European cosmetic regulation or local regulation, for example. We are based in France, near Paris, with a new lab since 2024 with 800 square meters only dedicated to sun protection testing. And so our approach is a global approach because we are not only a testing laboratory, we are also a provider of equipment, a provider of consumable for testing protocols, reference standards, for example, and we also provide training for customers and equipment calibration, and we are also able to help consumers, brands and consumers in terms of understanding the science behind sun protection testing. So our main activity is in vitro testing. We are primarily known as an in vitro sun protection testing lab because in the past we involved a lot in this field because we believe that in vitro tests were more ethical, with no need to expose human volunteers to UV, and also that they had the potential to be more reliable and repeatable than in vivo studies. So we have a very important catalog of in vitro studies for any needs that the customer can have in their product development or commercialization. We have screening tests, for example, to assess prototypes with SPF protection, UV APF, water resistance, or photostability. We have official claiming tests for the official norms, the ISO norm, for example, the FDA broad spectrum, the Boot Star rating, all kind of in vitro testing methods related to sun protection. And in addition to that, we have also many different specific claiming tests for specific claims, like, for example, blue light protection, sweat resistance, long lasting, ultra long UVA, etc. Each one of these tests was developed with an internal research study that is published in scientific journals. So when you test for these claims, you are sure that you have a reliable method to substantiate your claim. And we have also control testing for reformulation or comparison between different batches, for example. We also have the possibility to provide HDRS testing according to the new ISO 23698. So it is the hybrid method that combines in vivo measurements, non-invasive, and in vitro measurements to assess SPF, UVAPF, and critical wavelengths. And it's a very interesting clinical method for sun protection as an alternative to classical sun protection testing that irradiates volunteer under UV light. Here you have a table to have a recap regarding the regulation for sun protection testing, because nowadays there is no harmonized worldwide regulation. So every part of the world, every country, every area may have a different regulation. For example, in Europe, we can see that we rely on the ISO method with, for example, the 2 new ISO methods that were published in 2024, the in vitro SPF with ISO 23675 and the hybrid method with ISO 23698. In USA, you can only claim your sun protection with the FDA methodologies, for example. And in other areas of the world, you can have specificities. For example, in Japan, the in vitro UVAPF is not allowed, only the in vivo UVAPF. Whereas in Australia, it's the contrary. You can only claim your UVAPF in vitro and not in vivo. So it's the kind of thing you must know when you have to commercialize a product and have the regulatory dossier. We are also an important provider of equipment for sun protection testing in accordance with the ISO methodologies. So we can commercialize the substrates that are used for in vitro sun protection testing. So the substrates where the products are applied, spread before having measurements and a protocol testing methodology in order to give you the SPF result or the UVAPF result. And we provide these substrates that are nowadays the only substrate to be fully compliant with all the in vitro sun protection methods. We also provide equipment to improve the testing process with the spread master for robotic spreading. That is mandatory, for example, in the ISO 23675 for the in vitro SPF. And we also provide TherMaster, which is for temperature control, because during tests, some products can be very thermosensitive with variations in results due to temperature variation. So it's very important to have a very well-controlled temperature, and this appliance will help to have a better control of temperature. We are also a distributor of solar light for other kinds of equipment, such as spectrophotometers, solar simulator, and DRS devices for HDRS method. We are distributors of solar light for the European market only. So if you are based in the US and you need such equipment, you can contact Solar Light directly. For our own equipment, Spreadmaster, TherMaster, and PMMA plate, we are distributed in the US by LabSphere and Solar Light. So if you have any requests related to equipment, don't hesitate to contact them for quotation or further information. And also one important activity we have is the training part in order to train customers, whether they are testing laboratories, cosmetic brands, manufacturers, and if they want to learn how sunscreen testing works, how to perform sunscreen testing in good condition according to the ISO. So we have different kind of modules depending on the need and the level of knowledge of the customer. For module 1, for example, which is a very general introduction to sun protection concept. And then module 2, which is more a theoretical module to go deeper into the details of the ISO methods for in vitro or in vivo tests. And then we have module 3 with deep theory and also practice part. So people can go in our laboratory to have practical training, or we can send the technician to the customer's lab to have this kind of deep training. So here you have the contact information for us and for our US distributors. So if you have any question or anything you want to discuss, don't hesitate to reach us.
Thank you very much, Guillaume, for this very global presentation on this specific table of what are the guidelines for UV testing, and it's always tricky to know where it is in the world. So thank you very much. Thank you also for the timing, all the participants. Now I introduce Arthur. We keep the best for the last presentation. Arthur is the co-founder of Loretta, and he will present this innovation related to skin evaluation. Thank you, Arthur.
Thank you very much, Anne, for the introduction. Here is my screen.
Yes.
So I will present to you how at Loretta we are using AFM, so physician technology, to evaluate the skin and other type of sample. So for those who don't especially know much more about AFM, it's atomic force microscope. You can imagine that it's a needle that will go through your sample and draw the topographical map or measure the viscoelastic properties of your sample. It was mostly used in nanomaterial field, but with the new Loretta's AFM, we are able to analyze a lot of biological sample, like for example, skin cells corneocyte. So we will obtain a topographical map of your corneocyte from 3 micron to 50 micron with a really good resolution and also a super fast analysis. The advantage of atomic force microscope are multiple. The first one is that we don't look at the cells, we directly feel them. So we are independent from the skin color or the age, for example. So that means we are able to evaluate the severity of disease or the efficacy of cosmetic product on every type of skin. Also, to analyze the skin, we are using tape stripping. So it's a really easy method that can be performed by anyone to collect the sample. It's non-invasive, and also the quality of the measurement, the topography of the cells is not changing with temperature and time. So we are able to analyze old 1 year old sample with exactly the same precision over the result and the same result, and without any -80 refrigeration. So let me show you more in detail what it looks like when we analyze skin with atomic force microscope. What you can see there, those orange square scans, are the AFM scan. Those are 20 by 20 micron. It's around half of one cell. Here we are only looking at the cellular matrix of one cell. And we will see how the topography of the cell, the corneocyte, will change with the application of cosmetic product or severity of disease or treatment. You can see on the damaged skin that we have a lot of small dots everywhere. It's creating those really little circular defect. Those ones are called CNO, circular nano object, and they are a biomarker of the skin health and of the skin barrier function. They are directly related to things like natural moisturizing factor, lipid structure, hydration of the skin, and skin barrier compromisation. And when we will study the efficacy of a cosmetic product or the severity of treatment, we will see how the skin is going from a damaged, topographical look-like cells to a really healthy look-like cells. So we will count those number of CNO to get strong quantitative data to evaluate the efficacy of the product. And we will also look at really other parameters like the rugosity of the cells, the contrast in the height, a lot of physics parameter, but that provide a lot of information about how the cell is feeling. Is it like stressed? They have problem of hydration or barrier function. To show you some example of results, here you have some dermatology, dermocosmetics, and cosmetics result. We have applied this technique over a lot of dermatological disease. One of them is atopic dermatitis. With the CNO number and their density, ECTI, we are able to detect if we are on a healthy zone, non-lesional area, or lesional area. We can compare the different severity of the disease and with a really good sensitivity and strong statistical data. Also, something interesting is that because, as I said before, we are touching the skin and not seeing, we are able to analyze every Fitzpatrick with the same quality. So we are able to evaluate the severity of atopic dermatitis even on the really dark skin. For more cosmetic application, here you have an example where we have tried over 30 student hydration lotion, hydrating serum, and tonic regulator of pH. And we have observed a decrease in the number of CNO over all the students, up to 23% of decrease. So that's how we are able to say, okay, the product is efficient to restore the skin barrier, to hydrate the skin with this amount of percent through the method of CNO. So that's how we are working with corneocyte, but we also have other type of sample that we can analyze with AFM, because AFM is topography, so there is other type of sample like skin explant. Here we have cuts of skin explant, and we can look directly at the collagen fiber. If we are studying product for more anti-aging effect and want to see their effect on the collagen fiber, the quality of the collagen fiber, we can use AFM on cut of skin explant to evaluate the fiber size, the collagen fiber ridge regularity, and even the fiber alignment. To perform this, we'll make multiple scans from different sizes, 20 by 20 micron, 10 by 10 micron, and 5 by 5 micron. And with the high resolution, we can obtain good image of the ridges and the collagen fiber to evaluate their alignment, their quality, and their regularity in the ridges. Finally, another type of sample that we can analyze that is not for skin, but that is super interesting in cosmetics is the hair analysis. We can study hair cuticle with the AFM, the exact same AFM, so the same speed and the same resolution. And we will evaluate the roughness of the hair cuticle, the regularity of the edge of the cuticle, the lift of the cuticle, but also if there is some high roughness area, if there is some hole in the cuticle, some ghost cuticle, a lot of things that we can observe through AFM imaging over the sample. And you can see here the big difference between a healthy hair and a damaged hair with high roughness and low regularity. To show you some example of hair study, you can see here how her hair can evolve with a cosmetic product or hair treatment from T0 to 28 days. You can see that at the beginning you have really high roughness and low regularity, and at the end you have much better roughness and regularity. To combine and to produce this analysis with your other method and to present it to any client or any collaborator that you get, because we have a method that is touching and not seeing, we like also to be able to touch our sample. So we will 3D print the different type of sample that we are analyzing. So you can have really a 3D representation of corneocyte, hair fiber, and really other type of sample. So to resume, with the AFM, you can have a high precision analysis over only one scale, or on the collagen fiber and hair. You have an all-inclusive biometric analysis because we can analyze any skin color, any age, any condition, and a lot of type of biological sample. And that's a kind of analysis that is super easy to perform with only tape stripping for the classical corneocyte analysis. And finally, with the really strong visual power of our analysis and the 3D printing of the different type of sample we obtained, you can have clear number and striking image for marketing and for the educational purpose of your results. So thanks for your listening, and thank a lot, Anne, for the invitation.
Thank you very much, Arthur, for your presentation. Very clear, and every time I watch your one of your presentation, I learn something. Thank you very much. So we will finish this webinar with the presentation of SkinOps, and this time Ilona is joining us to introduce the beauty trends, and then I will go through the other one.
Hello everyone, so I'm Ilona and I'm really thrilled to be here today. I'm the communication manager at SkinHubs, and so today we will introduce first of all the North American beauty trends. Then we will talk about the testing exhibitors that were present during NYSCC Suppliers Day, and then Anne will go through the key testing trends from the show and the USA. So regarding the North American beauty trends, what we're seeing today is that the US beauty and personal market is one of the most dynamic in the world, with about $124 billion in 2024. And skincare is still the largest segment, with $28 billion in 2024 also. We're seeing shifts for the consumers because they are paying more attention to scientific validation. In a study that we found, 80% of consumers were saying that scientific validation had a direct influence for their purchasing choice. And 39% of them are very interested in skincare that are derived from clinical research. So what we're seeing is that consumers are more informed and also are looking for something more scientific, and we can say the science behind the marketing claims. And we're also seeing that for 85% of them, they are ranking quality and efficacy above price, reviews, and even brand heritage, which is the story of the brand. And for Gen Z and young millennials, they are willing to pay more for the products if there are science behind it. And we've also been seeing three major trends for the consumers, which is well-being and neurocosmetics. About 60% of women in the US are prioritizing well-being and mental health in their beauty routine. We are also seeing a rise of skinification, which is considering hair care like the skin. And for now, Gen Z, for 60% of them, they are using scalp-specific products, which is something very new on the market, and we're seeing a rise of this. And I think Biome Labs introduced this concept very well, which is the microbiome. And for 2027, the forecast is about $3.5 billion in the world, so it's a dynamic market. And I'm letting the floor to Anne.
Okay, so let's go through the testing. So first of all, at New York Suppliers Day, what were the exhibitors? So we see here the list of the clinical testing laboratories and the ones that are doing preclinical only and regulatory, and the other one preclinical and clinical, and also the instrumentations. I just remind you that the presentation will be on replay on the SkinHubs YouTube channel and the PDF also, so you can have this information when you like. Then here we see you see there is an increase of the number of the exhibitors regarding testing. There were 55 companies, labs, and instrumentation in 25, and in 26 there were 64. This is the distribution between preclinical and the several categories, and here the distribution per territory, and we see that North America is represent half of the exhibitors, and Europe the main. Yeah. So these are our valuable co-exhibitors and sponsors of the We Are Testing booth at New York NYSCC, because this is our collaborative way to share the innovation, to share information about testing. So there were CIDP, Nevisance, Biome Labs, Wineos, and Loretta who present their novelties. This is what we found about the testing lab news. So Biome Labs with spoke about microbiome and their new in vivo testing, and CIDP spoke about pigmentation, but they also have a big services, testing services about for hair care, sun care, or tolerance. Then we saw CompLife with transcriptomic insight and advanced testing model for preventing skincare. Elide from Korea, specifically a new test for oral care and laser Doppler on gum. And then Hellobiome, also from microbiome, Kind2Biome, also for analysis of the microbiome, Life Analytics Biobasic with a full service testing, Lipotype, I think they were new in New York, they are from Germany, and they do lipids meta-analysis for all claims, and Loretta. And SGS developed a new instrument, a new method using for the hydration and barrier function using Oriba instrument. Revivo Biosystem were present also at New York. Validated claim support with their specificity on photo before and after and biometeorology. They are located in New Jersey. And Wineos, Guillaume told us their expertise, and Zurko with their new hair care system with the Volscan profiler. These are the instrumentation news. We did have information about CBAS. C&K presented the new Cliniscale. It's tool, Nomad tool for hydration measurement, and Cortex present also the ultrasound. EOTech present the mini surf, and Loretta and Grandfield their new system for 3D imaging. These are the key testing trends now. What we see, I really like this matrix because it's a way to understand how the field of testing is influenced by many parameters, and this one has really changed comparing to the one from last year. And what we see is that the economical is increasing, and with the US politics, but not only, because I wanted to introduce the China, Japanese and Korean beauty, which is really giving a pressure on the market, on the global market, for the testing. So that's very interesting to see. So what increased also everything about microbiome. It was on all the lips since a few years, but now it's really a trend, and there are many actors, and we will see that in details later. And testing technology also, it's very impressive how it impacts the testing protocol and the studies, and with the biomarkers, with the AI, it's very important. The digital experience, nothing very specific to say because it's constant. Lifestyle changes. Inclusivity is very specific for the North American territory. Longevity, it's also interesting. And what we see, no evolution on the formulation, and beauty cultural is increasing. As Ilona said, now science-based and like efficacy, it's the second criteria of purchase, and this is really specific for North America because it's really increasing. Maybe it was more common in Europe, more common in Japan, but in US it's really a big trend. So this is the Skinos barometer, and I just focus for the factors that influencing the testing market. For US, it's in blue, but we already spoke about that. This is about the testing claims. Most claims per territory, and you see that whitening or multifunctional, it's very important. Moisturizing, but it's everywhere. And microgram, it's the territory where microgram is more important. Longevity also, and inclusivity, as I said, and also acne. Here we see the testing science routine in terms of technology, and what we see, it's the most important in North America is the photo before after. It's really a tradition of marketing to see directly for the consumer the effect of the product before and after. But now it's more with HD, which HD images and which AI treatment and algorithms, but it's still very important. Hair evaluation, it's also important, but and size base is it's increasing, and you see consumer test. It's very interesting, and there were only 6 zeros dedicated to only clinical, and on these 6, I think there were 4 or 5 just dedicated to consumer testing. So now this is the trends. I want to say something about neurocosmetic trends and well-being aging, which is really a trend in US and North America, with the holistic way to evaluate the importance of skin and mind, the multiparametric, and also the technology. And it's like there is a new skin barrier consideration, and these are the several methods we can use, and always you can find all this technology on the testing platform. Really easy to use. You can filter by instrument, by country, by claim, by whatever you like. So don't hesitate. And here about hair skinification, what we see, there is a micropatternic 3D human hair, and I suggest you to have a look at the ELID protocol. It's very, very interesting. We can also look at the surface of the hair, like Arthur proposed, on the electron microscopy or the Minister from EOTech. So there are many, many ways to evaluate skincare performance. Regarding microbiome, it represents a market of 835 millions. And in US it's 81 million, so it's 10% of the world. And you have key players, and you have also impact of the microbiome. Now we are looking at anti-aging, anti-acne, of course, but cleansing and moisturizing. And at the moment there is no official regulatory recognition, and labels are only issued by private organizations. And here, it's just a remember. I don't want to do too long. And this is the in vitro method. It can give a narrow view, but it's controlled and reproducible. And when we test the microbiome, we can use biopsy, stripping, swabbing, atom kits. That's the one from Biome Labs, and it's broad view, but it's high interpersonal variability. And then I want to focus to compare in vitro and subject. And subject, you use skin swabbing, and you can use at home. And this is a reminder about genomic, transcriptomic, and proteomics. And now the goal is really to study with shotgun or CSS or all the omics technology to see how it works. What are the metabolites? Which metabolites are active? And don't hesitate to collect the white paper on the SkinOps platform of the Cosmetic Testing News. In USA, what we have on the platform, you can find 200 for in vivo clinical. It includes sensory, consumer test, hair care, and also test on hair strands. You find 230 methods, 48 testing providers, and 38 instrumentation makers. And for in vitro, there are more than 300 methods and 54 providers. So if you want to stay tuned, there is all webinars are on the Skin of YouTube channel. You have the cosmetic testing news. If you are not, you did not subscribe already, you can subscribe to the weekly newsletter. And also, please save the date for the Cosmeto test in June 2-3 next year, because this year it was in March, and we gather 2 days of academic and high level of scientific information about testing. This year we were 270 people, and with remote possibility. Thank you very much for your for listening to this webinar. If you want to join We Are Testing, don't hesitate. And now we will have a look to the questions. Please, Renato and all participants, please join me, and we will have a look to the question. So, there were questions about microbiome. There were questions about pigmentation. I think Renato already answered. So, I can't do anything. My system is blocked. I don't know why. Tu vois, regarde, ça bloque là.
Clique sur Zoom.
Voilà, okay. Sorry. Yeah, sometimes I need some support for the. So we have a question about the pigmentation, but it was really specific to the pigmentation. I don't know if you have. I am a little bit lost in the. Okay, so at the moment, I think that the question was answered about there were questions about the microbiome. There were questions about the AIS from Jonas. Jonas, would you like to say something complementary?
It was a question there, and I respond to that. But that EIS does not measure biomarker directly, such collagen and things like that. Instead, it measures the changes in the skin structure, hydration, and barrier function, and that can be influenced by these markers. So I have responded to the question in the.
Okay, great. And Guillaume, would you like to add a commentary to comments to the question of Jorge regarding in vitro methodologies?
Yes, small insight, because it's a relevant question, the correlation between in vitro and in vivo SPF. You have to know that in the past, like 20 years ago, in vitro tests were not as reliable as they are today. 20 years ago, there was no control of the substrate, no control of the parameters, the temperature, the radiation, for example. So over the years at Wineos, we invested a lot in research to improve the in vitro methodologies with many research studies about the impact of spreading, for example, the impact of temperature, the impact of UV exposure of the substrate. And so it took years, but in vitro methodologies were improved, also through the regulation bodies like AFNOR, Cosmetics Europe, and ISO. And nowadays, we have a very complete method with the in vitro SPF ISO 23675, which was demonstrated to be correlated with the in vivo SPF by ISO 24444. So sometimes people still doubt about the correlation, but the facts are here. There were many interlaboratory studies with ring tests to compare the result between in vitro SPF and in vivo SPF. And so the correlation was demonstrated on several products with different kinds of product, different kinds of filter. So nowadays, there should be no doubt about the correlation because, for example, ISO 23675 is nowadays the in vitro SPF method with the most published data, and especially published data in peer-reviewed journals. So it's really a demonstrated correlation to answer the question.
Thank you. Just to finish, because I think we are now out of the time, but Arthur, would you like to say something about the question you had?
Yeah, I would just complete something. The question was how to be sure and how to ensure that iteration level of the content will not modify the IFM measurement after the collect. The idea is that depending on what kind of AFM measurement you're doing, if you are studying viscoelastic properties of the corneocyte, it's super important to keep the iteration and the humidity level at a good level. But when you are studying topography, the topography is not changing after the collection, so you just have to keep your corneocyte, your tape-stripped corneocyte in the Eppendorf, and that's enough to ensure proper results. That's all.
Okay. Good. Thank you very much, everyone. Every presentation will be available. We will send you an email specifically, and the webinar will be online. I hope you enjoyed this webinar and hope to see you soon. We have a webinar dedicated to hydration measurement on the 30th of June. See you soon. Bye-bye. Have a good day.
Bye.
Bye-bye.
Bye, thank you.
Bye-bye. Thank you, Han.
Bye-bye. Thank you.